Okay, so everyone, welcome to this seminar series newly named as the NextGen Precision Health Seminar, Neuroscience Seminar Series. I really appreciate today's Dr. Russell Swerdlow from KU Medical Center to present his own research. And Dr. Russell Swerdlow is a professor in the Department of Neurology, Molecular and Integrative Physiology, Biochemistry, and Molecular Biology at the KU Medical School. He directed the KU Alzheimer Disease Center and is a neurodegenerative disorder program, as well as the Heartland Center for Mitochondrial Medicine. So we are really enjoy to join their group for scientific exchange. And he received undergrad and MD degree from New York University and trained as a neurologist and Alzheimer's specialist at the University of Virginia. He holds the gene emergent Sweeney Chair at the KU and the recipients of the Weir Marshall Award from the American Academy of Neurology, recipients of the Weir American Parkinson's Disease Association, and the Chancellor's Club Research Award from the KU. He currently sits at the NIH Board of the Scientific Counselor, and Dr. Swerdlow's research focus on brain energy and metabolism. His roles in the Alzheimer's disease and the therapeutic manipulation. Welcome, Dr. Swerdlow, and the time given to you.
Okay. Well, thank you very much for that kind introduction. And for the next 50 minutes or so, I'm going to talk about why I think mitochondria are important in Alzheimer's disease and why they may even play a primary role in Alzheimer's disease. Certainly, it's been recognized for quite a while that mitochondria are altered in people with Alzheimer's disease. The mitochondria in the brains of people with Alzheimer's disease look different. There's a shift towards mitochondrial fission. The process through which new mitochondria are generated, mitochondrial biogenesis, and through which old mitochondria are removed, called mitophagy, those processes are reduced. There are reductions in enzyme activities, including for an enzyme called cytochrome oxidase, or complex 4 of the respiratory chain, which I'm going to talk about later. And there are changes in mitochondrial DNA. So there are variants, population variants in mitochondrial DNA that appear to influence lifetime risk of developing Alzheimer's disease. There are increases in somatic mutations of the mitochondrial DNA, be they point mutations or deletions. There is increased oxidative damage to the mitochondrial DNA. And there is actually less mitochondrial DNA present. And I'm going to circle back to that in a little bit. It's been observed now for about 15 years that beta amyloid also co-localizes with mitochondria, and either through this co-localization or through indirect effects may alter things like oxidative stress and cause reactive oxygen species production, or alter calcium homeostasis, and that this ultimately may lead to respiratory chain failure and cause lots of other downstream problems. And this is what I would refer to as a secondary mitochondrial cascade. It fits within the view of the amyloid cascade hypothesis, and may in fact explain, if this hypothesis is correct, how does it cause downstream damage. But there are some problems with this view because let's see here. Yeah, some of these changes are not brain limited. So there are mitochondrial changes in platelet mitochondria and lymphocyte mitochondria fibroblasts, and in muscle. So in these cases, it kind of becomes hard to attribute all the changes in mitochondrial function and viability to beta amyloid. And because of this, I've proposed what I would refer to as a primary mitochondrial cascade hypothesis, which proposes that we inherit a baseline mitochondrial function and baseline mitochondrial biology that comes through genes we inherit from our fathers and from our mothers, the nuclear genes, but also the mitochondrial DNA that comes solely from our mothers. And that over time, it's generally well accepted that mitochondrial function tends to decline, and this can be associated or perhaps due to changes in mitochondrial DNA and reductions in the mitochondrial DNA copy number. And where this primary mitochondrial cascade hypothesis really differs from, let's say, a secondary mitochondrial cascade hypothesis is that I'm proposing that these mitochondrial changes may then lead to the proteostasis changes, the plaques and the tangles that we associate with Alzheimer's disease, and with the systems changes that we increasingly recognize as being associated with Alzheimer's, such as insulin resistance, altered brain perfusion, microvascular changes, altered lipid biology, and microglial activation and inflammation. And perhaps there is a feed-forward phenomenon in which these proteostasis changes and these systems changes may further impair mitochondrial function, but certainly over time, just on its own, I would expect that mitochondrial function would continue to decline, and eventually a point would be reached at which the brain simply could not compensate anymore for this. And at that point, you would transition from compensated brain aging to uncompensated brain aging, and this would result in the progressive cognitive decline and dementia that we associate with Alzheimer's. And one of the reasons why I began to think this way has to do with studies that I performed as a postdoctoral fellow in a mitochondrial lab. The project I was working on was trying to determine why cytochrome oxidase activity is systemically reduced in people with Alzheimer's disease. So here is the structure of cytochrome oxidase, a multimeric holoenzyme in which 10 subunits are encoded by nuclear DNA, and 3 of the subunits are encoded from mitochondrial DNA, which I'm showing here. You can see in red, cytochrome oxidase subunit 1, subunit 2, and subunit 3. So the hypothesis that we were trying to address was whether mitochondrial DNA might be contributing to this reduction in cytochrome oxidase activity in the Alzheimer's patients. And the way we studied this back in the 1990s was by adapting what was then a recently described technique called the cytoplasmic hybrid or CYBRD technique, which I'll walk you through now. It's really not that complicated. So you can take, what you do is you take a cell line. For example, we like to use neuronal cell lines. So a lot of our experiments have used human SHSY5Y neuroblastoma cells. And I've shown here a cartoon of a cell with a nucleus, and the nuclear encoded respiratory chain subunits are shown here in blue. And there's mitochondrial DNA in the mitochondria, which codes respiratory chain subunits, which are shown in red. And all these subunits coalesce on the inner mitochondrial membrane to make a functional respiratory chain. And it's possible to completely remove or deplete the mitochondrial DNA from these cells. When you do that, the cell is now something called a Rho zero cell. And the reason why it's called a Rho zero cell is that Rho DNA is another word for mitochondrial DNA. So no mitochondrial DNA equals no Rho DNA or a Rho zero cell. And of course, in that situation, you no longer can express the mitochondrial DNA encoded subunits of the respiratory chain. So what you're left with is a cell that is no longer respiratory competent. And it's possible to keep these cells living and to even expand them by adding the proper nutritional support into the culture medium. And then one thing you can do is you can draw a tube of blood from an individual, and you can centrifuge out the platelets. Platelets are great for these experiments because they don't have nuclei, they don't have nuclear DNA, but they have mitochondria in them. And you can add into a tube Rho zero cells with platelets, and you can add a detergent to rough up the membranes and mix them. And when you quench the detergent, when the membranes reform, in some cases, you'll have cells that have mixed the cytosols of the platelets and the row zero cells, which are now no longer row zero cells, because you can see here that there's now mitochondrial DNA in the cell. It came from the donor. And over time, this transferred mitochondrial DNA will repopulate the cell's mitochondrial DNA and restore respiratory chain function. And so what you can do is you can generate, let's say, a series of cybrid cell lines from people with a particular condition, let's say Alzheimer's disease. So you make a bunch of Alzheimer's cybrid cell lines, and you can make a series of cybrid cell lines that contain mitochondrial DNA from age-matched control subjects. And then what you can do is you can subject these different cell lines to molecular and biochemical assays. And if there are any functional differences between these groups of cell lines, it presumably reflects differences in the mitochondrial DNA between the cell lines. The reason is that the nuclei is the same between all the cell lines, as is the environment that you're growing the cell lines in. So if there are functional differences in the cell lines, that's probably reflective of differences in mitochondrial DNA. In fact, we did find that when we transferred mitochondrial DNA from people with Alzheimer's disease to make these cybrid cell lines, that we succeeded in transferring, at least in part, the cytochrome oxidase defect that we see in the Alzheimer's patients. And we saw other differences as well. For example, we saw these misshapen mitochondria with disrupted cristae in the Alzheimer's cybrid cell lines. The Alzheimer's cybrid cell lines had increased amounts of superoxide production, so increased oxidative stress production. And 25 years later, we're still not entirely sure of what distinct mitochondrial DNA features are accounting for this, but we recently published that there actually is a reduction in the mitochondrial DNA copy number in the Alzheimer's cybrid cell lines versus the control cybrid cell lines. Well, we've been very interested in trying to link these primary mitochondrial defects to the classic histopathologies of Alzheimer's disease, such as to the tau and the tangles of Alzheimer's. And just by way of background, tau is a protein that normally binds to microtubules. And there are different tau isoforms that are dependent on transcript splicing. And when we're forming in utero, our tau is heavily phosphorylated. And when we're adults, this phosphorylation is lost. But in those with Alzheimer's disease, phosphorylation returns to the tau. And phosphorylated tau does not bind well to microtubules. In fact, that tau then begins to oligomerize and to form fibrils, and these fibrils are what form the tangles. And tau is used as an Alzheimer's disease biomarker. It's believed or assumed that tau protein is released when neurons die. And certainly it is known that tau is actively secreted by cells. And because of either one or both of these reasons, presumably, spinal fluid and plasma phosphorylated tau levels are increased in people with Alzheimer's disease. And in spinal fluid, total tau levels are increased in Alzheimer's patients in the spinal fluid and in multiple diseases and in fact, after traumatic brain injury. And we recently reported that if you measure levels of oligomerized tau in Alzheimer's cybrid cell lines, you see that there are increases in the amount of oligomer tau or pre-tangled tau in the Alzheimer's cybrid cell lines versus the control cybrid cell lines. And we wanted to look more into this, how primary mitochondrial defects alter tau biology. And I'm going to switch now to showing you data that were generated not from studies of cybrid cell lines, but by just looking at how tau is being handled in row zero cells. And the reason why we did this is that it's a lot easier to do these mechanistic experiments when we only have two cell lines to handle, the wild type SY5Y neuronal line and the SY5Y row zero line. That's much easier to handle than having 24 or more different cybrid cell lines. And what you can see here, this is a Western blot of tau protein from wild SY5Y cells and SY5Y row 0 cells. You can see here in the wild type cells, there's one predominant tau band. And if you look over here at the tau in the row 0 cells, well, there's not just one band, there's more of a smear. And there's a lot more tau. And then you see this high molecular weight band here, which actually turns out to be tau dimer. And if you clean up the protein lysate with a phosphatase, now in the row 0 cell, you can now see the distinct different isoforms that are now being expressed much more heavily in the row 0 cells. Again, you see that there's much more tau, and there's this tau dimer up here. And when we did dot blots using an antibody that detects only tau oligomers, we saw that there was this robust increase in tau oligomer in the row 0 cells. And when we did partial mitochondrial DNA depletion by treating SY5Y cells with ethidium bromide, which intercalates into DNA and it blocks mitochondrial DNA replication. After about 1 day of treatment, there was about a 50% mitochondrial DNA depletion. After 3 days, it was about 70%. And then after a week, it was about 95% mitochondrial DNA depletion. And you can see here that as we deplete the mitochondrial DNA, the tau begins to oligomerize within the cells. But what about amyloid precursor protein and beta amyloid? Well, by way of background, the APP gene has been implicated in Alzheimer's disease through genetic studies. The APP mutations in the APP gene are rare causes of autosomal dominant Alzheimer's disease. And there is even an APP population polymorphism that actually acts to reduce one's lifetime Alzheimer's risk. Mutations in two presenilin genes, presenilin 1 and 2, will also lead to autosomal dominant Alzheimer's. And this is relevant because the presenilin proteins play a role in APP processing. But despite recognizing all of this, APP functions are incompletely understood. What about beta amyloid? So A-beta has been tied to Alzheimer's disease through plaques. It's been proposed to cause Alzheimer's disease. This really is the basis of the amyloid cascade hypothesis. And the dominance of the amyloid hypothesis has caused, on the clinical front, changes to the definition of what Alzheimer's disease is. So, for example, in 2021, you can have plaques in your brain, but no tangles and no cognitive changes. And that will qualify you for a designation of something called Alzheimer's pathological change. And A-beta also serves as a basis of an Alzheimer's biomarker. In spinal fluid and plasma, levels of the 42 amino acid A-beta 42, and the 40 amino acid A-beta derivative A-beta 40 they're both reduced in spinal fluid and plasma of Alzheimer's patients. And the A-beta is reduced to a greater extent so that you see reductions in the A-beta 42 to A-beta 40 ratio. And sort of a popular assumption, I would call it an assumption of why this is that neurons are consistently secreting A-beta 42 and A-beta 40. And the idea is that perhaps if there are plaques present, that some of the secreted A-beta 42 and 40 glom onto the plaques. And because A-beta is less soluble than A-beta 40, the A-beta 42 is less soluble than A-beta 40, the idea is that perhaps more A-beta 42 gloms onto the plaque, and that leads to a reduction in the A-beta 42 to A-beta 40 ratio in the spinal fluid. Well, we've done studies before in Alzheimer's hybrids looking at beta amyloid levels, and these are data that we previously reported showing that in cell lysates of Alzheimer's hybrid cell lines, that they contain more A-beta 40 and more A-beta 42 than control hybrid cell lines. And we more recently have been doing a lot more experiments with how mitochondrial biology affects APP biology and A-beta. And we've done these studies in row 0 cells. So I'm going to show you now some data looking at how mitochondrial DNA depletion affects APP and A beta biology. So one thing we found is that the Rho-0 cells show a robust increase in the amount of APP mRNA. So APP gene expression is being turned on. And this spills over into having more APP protein within the cells. So this is APP protein, not A beta protein. And where's that APP protein going? Well, you know, we've done immunohistochemistry to try and determine this. And you can see here that you know, the APP is sort of staining maroon here. You can see that, first of all, there's more APP in the Rho-0 cells and the SY5Y cells consistent with the Western blots. And this APP is present in a granular pattern, which makes you wonder, is it co-localizing with mitochondria? In fact, it is. And how much APP localizes to mitochondria has a relationship with the mitochondrial membrane potential, with how active the mitochondria are from a respiratory perspective. So here we've stained cells with a dye called TMRE, which when mitochondria are more polarized, more TMRE will be taken up into the cells, and you'll see more red fluorescence. And when the mitochondria are depolarized or less active, the mitochondria will take up less of the dye, and you'll see less red. So you can see here in Rho-0 cells, there's less red, there's less TMRE in the mitochondria than there are in the SY5Y cells. So the Rho-0 cells are relatively depolarized, their mitochondria are anyway. And you can see here that there's more APP in their mitochondria. And similarly, if we take SY5Y cells and we depolarize them chemically by exposing them to a chemical uncoupler called FCCP, you can see here that leads to depolarization of the mitochondria and a routing of APP to the mitochondria. Conversely, if we hyperpolarize the mitochondria by treating them with a chemical called oligomycin, which is an ATP synthase inhibitor, when we hyperpolarize the mitochondria, which you can see here because there's more red, now there is actually routing of APP away from the mitochondria. And similarly, if we hyperpolarize the mitochondria by glucose starvation, just by taking the glucose out of the medium, which forces the cells to rely pretty much entirely on respiration to make their ATP, you can see that the mitochondria hyperpolarize, and there's a routing of APP away from the mitochondria. So I've been telling you now about what happens to APP, but what about A beta? What's going on with A beta during all of this? So here I'm showing you data from Rho-0 cells. And in this case, I'm showing you the amount of A beta protein that's being secreted into the medium. One thing you can see here is in the Rho-0 cells that they're actually secreting less A beta into the medium, both less A beta 40 and less A beta 42, and that's having an impact on the A beta 42 to 40 ratio. When we uncouple the mitochondria in SY5Y cells, when we depolarize them chemically with FCCP, again, we see that we're now that the cells are now secreting less A beta 40 and less A beta 42 into the medium, and now the A beta 42 to 40 ratio is actually lowered. Conversely, if we hyperpolarize the mitochondria with oligomycin, now we can see that the cells are secreting more A beta 42 into the medium and increasing the A beta 42 to 40 ratio. And if we glucose starve the cells and hyperpolarize the mitochondria, the cells begin to spit out more A beta 40 and A beta 42. We've extended these measurements to neurons generated from human-induced pluripotent stem cells that we've generated. And we've treated those iPSC-derived neurons with athenium bromide to deplete their mitochondrial DNA. And here you can see that we've managed to deplete about 60% of the mitochondrial DNA. And not surprisingly, the mitochondria are then depolarized. And you can see here that in these mitochondrial DNA depleted cells with the depolarized mitochondria, that these neurons are releasing less A beta 40 and less A beta 42 into the medium, just like what the case was with the SY5Y cells. And essentially what we're seeing is this relationship. When the mitochondria are depolarized, when the mitochondrial membrane potential is low, the cells are not releasing that much A beta 42 into the medium. But when the mitochondria are more active, when they're hyperpolarized, the cells are releasing more A beta 42 into the medium. Another relationship that we looked at is the relationship between mitochondrial APP and A beta secretion. And you can see here that when the mitochondria is localizing to when the APP is localizing to mitochondria, the cells are secreting less A beta 42. And when the mitochondria are not taking up a lot of APP, then the cells are releasing much more A beta 42. Similarly, when APP is localizing to the mitochondria, A beta 40 secretion levels are low, and when APP is not going to the mitochondria, A beta 40 secretion levels are elevated. And if you look at the ratio of A beta 42 to 40, when there's not a lot of APP going to the mitochondria, we see a low A beta 42 to A beta 40 ratio. And when only a little bit of APP is going to the mitochondria, we see a higher 42 to 40 ratio. Now, yeah, I'm going through this kind of fast, and I know it's a little bit confusing to track. So I tried to summarize all of this in this cartoon. This is what we think is going on. So here's a schematic neuron cartoon, and the cells produce APP, and the APP is transported to the synapse. And when mitochondria are active, when there's normal respiration, only a little bit of that APP gets siphoned off to the mitochondria. Most of it makes it to the synaptic membrane, and a lot of A beta ends up getting secreted. But if the mitochondria are struggling, if the mitochondria are abnormal, that APP gets siphoned off to the mitochondria, less APP makes it to the synaptic membrane, and there's less A beta secretion. What about mitochondrial biology and ApoE and apolipoprotein E? So the ApoE gene produces a protein, the ApoE protein or apolipoprotein E, which is a 299 amino acid protein. It forms lipoproteins with fatty acids and cholesterol. It's expressed primarily in hepatocytes and astrocytes, although stress neurons can also express it. There are three main ApoE isoforms, ApoE2, ApoE3, and ApoE4, which differ on the basis of what amino acids are present at positions 112 and 158. ApoE4, which is the ancestral human ApoE, has an arginine at both 112 and 158. ApoE3, which is currently the most common human isoform, has a cysteine at 112. And ApoE2, which evolved from ApoE3, has cysteines at both 112 and 158. And for unclear reasons, these isoforms associate with different Alzheimer's risk quotes. So those who have an ApoE4 have the highest lifetime risk of developing Alzheimer's disease. ApoE3 is judged to be a neutral risk, and those who have an ApoE2 actually have a reduced lifetime risk of developing Alzheimer's. There are other rarer isoforms as well, for example, the Christchurch variant of ApoE, in which upon an ApoE3 background, there is a substitution of a serine for an arginine at position 136. And people with the Christchurch variant also have a lower Alzheimer's risk. So there's something about getting rid of arginines that seems to lower Alzheimer's risk. And there is an association between ApoE protein and mitochondria, perhaps in the form of full-length ApoE, but certainly as a 272 amino acid ApoE derivative. What happens is, and this is really the work of Robert Maley, the discoverer of ApoE protein, and his colleagues, what they've published is that different ApoE protein encoded from different isoforms folds differently. So the ApoE protein has both a receptor binding domain and a lipid binding domain, and they're connected through this loop here, and that creates a hinge. And this hinge is swung open in ApoE3 encoded protein and in ApoE2 encoded protein. But in ApoE4, when you have this arginine at 112, it changes the folding of the protein, such as you form a salt bridge between two amino acids in the receptor binding domain and the lipid binding domain, and you pull this hinge in. And what that does is it sets up the ApoE protein to be processed by an as yet uncharacterized protease that cleaves off the 27 C-terminal amino acids. And when you do that, you open up an mitochondrial targeting site, and you get targeting of this product to the mitochondria. Well, like I said, we've been interested in how mitochondrial biology affects ApoE biology, and one thing we found, which was really quite striking, was in our row 0 cells, which don't have mitochondrial DNA, we see this massive upregulation of ApoE gene expression. So as it predicted, we really don't see much ApoE expression at all in normal neuronal cells, but in the mitochondrial DNA deprived cells, we see this almost 60-fold increase in mitochondrial ApoE expression. And that carries forward to more ApoE protein. Remember, ApoE is primarily a secreted protein. So we see a lot more ApoE protein now in the medium, the conditioned medium of row 0 cells. And if we achieve partial mitochondrial DNA depletion, the cells begin to turn on their ApoE expression. And we can it's kind of subtle, but we can even start to now see some intracellular ApoE in the cells with mitochondrial DNA depletion. Well, we've looked to see how different ApoE proteins might affect mitochondrial function and mitochondrial biology. And so what we did was we created SY5Y cell lines that are stably transfected to constitutively express either ApoE2, ApoE3, or ApoE4 protein. And we looked at certain biological phenomena, such as mitophagy, the removal of old mitochondrial DNA. And you can see here that in the cells that are expressing ApoE4, there are dramatic reductions in the proteins BNIP3 and NIX. These are proteins that play a role in mediating mitophagy, specifically a type of mitophagy called cell-mediated mitophagy. And over here, we transfected cells with a reporter construct that expresses both a mitochondrial targeted green fluorescent protein and red fluorescent protein. And when mitochondria are being removed through mitophagy, the acid from the lysosome that digests the mitochondria degrades the green fluorescent protein and leaves just the red fluorescent protein back or existing. So when you see red, that's a sign that mitochondria are undergoing mitophagy. And what you see here is that cells that express ApoE4 have less red. So this is consistent with less mitophagy flux. And we also transfected these cell lines with a reporter called mitotimer, which encodes a peptide that localizes to mitochondria. And it initially fluoresces green, but as mitochondria age, that fluorescence switches from green to red. So you can see here that the ApoE expressing cells have more red mitotimer in them. So again, consistent with reduced mitophagy flux. And when you measure cytochrome oxidase activity in these cell lines, COX activity is reduced in the ApoE4 expressing cells, which is consistent with what we see in other studies. So these are data that we recently published in which we measured platelet mitochondria cytochrome oxidase activity in individuals with Alzheimer's disease. And we divided the data up segregated the data by those but by their ApoE genotype. So these people have these people are E4 carriers, and these people are not. And you can see here that even though people with Alzheimer's disease have a lower cytochrome oxidase activity, it's even lower in those who are E4 carriers. Now, I've been talking a lot up to this point about how mitochondrial DNA may play a role in influencing mitochondrial function that influences Alzheimer's, and talking about how the ApoE gene may also play a role in affecting mitochondrial function and affecting Alzheimer's risk, which raises the question of, could there be a combination effect between mitochondrial DNA and ApoE isoforms? So these are data from two cohorts, the University of Kansas Alzheimer's Disease Center's clinical cohort and the Alzheimer's Disease Neuroimaging Initiative, or ADNI cohort. These are both longitudinal cohorts that follow people over time who are both cognitively normal and who have Alzheimer's disease. And it also ascertains, who converts from being normal to having Alzheimer's over the period of follow-up. And you can see here that those who have what's called mitochondrial DNA haplogroup J have Alzheimer's patients have an increased frequency of haplogroup J, mitochondrial DNA haplogroup J. And to some extent, this is being driven by people who also have an ApoE4. So it raises the question of, are you getting a double hit on the mitochondria? And these are data that we're currently developing through looking at the data in these cohorts. But I'll share them with you. So these are data from the KU clinical cohort. And we took all the people who, when they entered the study, were cognitively normal. And we tracked them over time. And over time, some of them start to develop Alzheimer's disease. These are older people. And you can see here in green, this green line, that the survival for getting Alzheimer's disease is not as good. So people with this green line are more likely to develop Alzheimer's over the years that we're following them. And this is the group of people who happen to have mitochondrial DNA haplogroup J and an E4 allele. And if you look at things in terms of function, this is again data from the KU clinical cohort. Annually, we rate them with a scale called the CDR-SB, or clinician or clinical dementia rating, some of the boxes score, in which higher scores capture worse overall function. And you can see here that that people who are haplogroup J, ApoE4 carriers, don't hold up as well globally as compared to everyone else. And if you look over here, if we track them with a scale called the mini mental state exam, or the MMSE, which is a measure of cognitive performance, you can see here that those who have a haplogroup J and an E4 allele don't hold up as well on this cognitive scale as compared to everyone else. And you see a similar pattern in the ADNI data. So those who have a haplogroup J and ApoE4, so over time show a greater decline in global function and a greater decline in their mini mental state exam scores as compared to the other genotypes. So I'm going to pivot now to talking about targeting mitochondria for therapeutic purposes in Alzheimer's disease. And it is my suspicion that we're already doing this to some extent. So the mainstay of Alzheimer's therapy in 2021 are cholinesterase inhibitors, which block the degradation of acetylcholine and increase cholinergic tone in the brain. And we've recently reported that using cholinesterase inhibitors increases mitochondrial respiration. And I think we know the mechanism through which this is working. I don't have time to go through the full mechanism, but just briefly, what appears to be happening is when you increase cholinergic signaling in neurons, you trigger an inositol triphosphate mediated calcium release at the endoplasmic reticulum. And this calcium that's released from the ER enters mitochondria and stimulates mitochondrial respiration. So I think to some extent, cholinesterase inhibitor therapy is actually a indirectly serving as a mitochondrial tonic. And it's been also proposed that perhaps we can promote mitochondrial biogenesis to treat Alzheimer's disease. And there have been some trials looking at this. So there are diabetes drugs in the class of the thiazolidine diones that can actually induce mitochondrial biogenesis. And there have been trials of thiazolidine dione drugs in Alzheimer's participants. The problem with these studies is that you can't get enough you can't give a person enough of the drug to get a brain level that will induce mitochondrial biogenesis in the brain, because these drugs really don't cross the blood-brain barrier. It's been proposed that perhaps we can promote mitophagy. This has actually become very popular recently. And there have been a number of papers in the last several years showing that if you give drugs that induce mitophagy, such as the antibiotic doxycycline, or the iron chelator deferriprone, or the pomegranate derivative urolithin A, if you give any of these drugs to transgenic mice that express a mutant APP or mutant presenilins, that not only do you attenuate the plaque deposition, you also preserve the behavior of the mice. It's been proposed that maybe we can protect mitochondria from things such as oxidative stress or ApoE or beta amyloid. And then finally, perhaps we can promote bioenergetic fluxes. And this is what I'm going to talk about for the next few minutes. So, we've certainly recently shown that you can affect brain bioenergetic fluxes pharmacologically. And we recently reported that if you give people high enough amounts of the Krebs cycle intermediate oxaloacetate, or OAA, that you can enhance brain bioenergetic flux. And then also we try to do this with ketogenic diets, which provide the brain with an alternative fuel source, ketone bodies, to metabolize. And this is preclinical work looking at how a ketogenic diet affects the brain of mice. So we did an experiment in which we placed mice on a ketogenic diet for 3 months. And at the end of the 3 months, we harvested their brains, and then we separated out their neurons and astrocytes. And we submitted the enriched neuron and astrocyte fractions for RNA-seq, for transcriptomic analysis. And we analyzed the data using multiple different software packages, such as KEGG, pathway analysis. And we looked at our KEGG pathway analysis data to see what diseases and conditions would be predicted to be affected by a ketogenic diet. And amazingly, the disease that rose to the top of the pack by far was actually Alzheimer's disease. And the reason why Alzheimer's disease was identified as a disorder that would be particularly impacted by a ketogenic diet is that in the brains of Alzheimer's patients, expression of genes that support the insulin signaling pathway and that support oxidative phosphorylation, that expression of those genes is reduced. But in the neurons of mice treated with a ketogenic diet, those neurons showed an upregulation of genes that subserve oxidative phosphorylation and an upregulation of genes that subserve the insulin signaling pathway. So the ketogenic diet is reversing RNA expression patterns that we see in the brains of Alzheimer's people. And that's why the ketogenic diet, it would be predicted to particularly affect Alzheimer's disease. This is what it looks like schematically. So each one of these rectangles here is indicating a gene. And when a particular gene is the expression of that gene is being picked up on the RNA-seq, you know whether gene expression is being increased, which shows up as red, or is being decreased, which shows up in blue. And each one of these, if you look at this rectangle here, each one of the columns in here are data from an individual mouse. And so you see, sometimes there's not much of a change. That would be in gray. Sometimes there's upregulation that's in red for that mouse, or blue that's downregulation for a particular mouse. And so, these experiments and with these biological systems are never really fully clean, but you can get a sense here that there's more red, there's more upregulation in complex 1 genes, here in complex 2 genes, complex 3 genes, complex 4 genes, cytochrome oxidase, and complex 5 genes. You see more red than blue. And because of this, the software interprets this as an upregulation of oxidative phosphorylation in the ketogenic, in the neurons from the ketogenic diet-fed mice. And this is the schematic for the insulin signaling pathway. Again, red is upregulation, blue is downregulation. And you can see here for any particular gene in the insulin signaling pathway that you're interested in, you can see that there tends to be more red than blue. And for that reason, the software interprets this as an upregulation of the insulin signaling pathway. Well, what if you actually give a ketogenic diet to people with Alzheimer's disease? Well, we've done that study. So I'm going to share with you data from a study. We performed a pilot study called the Ketogenic Diet Retention Feasibility Trial, or the K-Draft. And in this study, what we wanted to do was to enroll 15 individuals with Alzheimer's disease into a study in which they spend 3 months on a ketogenic diet supplemented by a medium chain triglyceride oil. And with ketosis monitoring and cognitive testing before people initiate the diet, after 3 months on the diet, and after a 1-month washout of being off the diet. And of the 15 people that we enrolled, 10 of them were able to implement the diet, 5 were not. Of the 10 people who were able to implement the diet, 9 were protocol compliant and 1 was not. And the protocol deviation had to do with the fact that one of the participants discontinued their Alzheimer's medications. So it was quite a bit of work to recruit for this. We had to pre-screen almost 300 people to recruit our 15 individuals. But the people that were in the study and were able to implement the diet did achieve ketosis. So this is data from daily urine ketone monitoring. And you can see here that some of the people were in ketosis most of the days of the study, and some of them were only in ketosis for some of the days. Some people had a relatively robust ketosis. Some had just a more mild trace ketosis. And when we measured it in the serum, you can see here that serum beta hydroxybutyrate levels were increased at months 2 and 1 after months 1, 2, and 3 overall. What about their cognitive performance? Well, the one participant, this was the protocol non-compliant individual, actually lost ground over the 3 months in the study, as shown on a scale called the ADAS-Cog, or the Alzheimer's Disease Assessment Scale, in which lower scores are better scores. So this is getting better and this is getting worse. One participant neither declined nor improved, but the other 8 participants all showed some degree of improvement on the ADAS-Cog. Some of them showed quite a bit of improvement on the ADAS-Cog. I mean, an improvement of 6 points or more on the ADAS-Cog, that's an improvement that you can really see from across the room. And if you bear down on the data from the individual subjects some more, you can see that we frequently saw this pattern in which people would enter the study with their baseline performance, as shown on the ADAS-Cog, and then after 3 months on the diet, their ADAS-Cog score improved, and then after a 1-month washout, their score reverted about to where they were at the start of the trial. Now, there have been other studies looking at the impact of ketogenic diets on cognition in people with Alzheimer's disease and with a condition called mild cognitive impairment, or MCI, which is a frequent precursor of Alzheimer's disease. So there's a study of Krikorian et al., which found that that individuals with MCI placed onto a low carbohydrate ketosis inducing diet showed cognitive test score improvements, and that this correlated with urine ketones. There's a study of Brandt et al., which included people with mild cognitive impairment and Alzheimer's. And they reported that among those who were able to implement a ketogenic diet, that those individuals showed improvements in their cognitive scores. There is a study of Neth et al., which enrolled people with MCI and also subjective cognitive impairment. So really cognitively normal people. And they measured biomarkers on the individuals, and they found that those who were, when they were on a modified Mediterranean ketogenic diet, that their spinal fluid biomarkers changed in directions that one would assume were beneficial changes. So the CSF-A beta levels increased and CSF tau levels decreased. And there was also some evidence of increased cerebral perfusion during the ketogenic diet. And lastly, the study of Phillips et al., which enrolled 26 Alzheimer's participants and reported improvements with the ketogenic diet in scores of daily function and quality of life, and then a strong trend towards cognitive improvement. Well, one of the limitations of all these studies is they're all small studies, not randomized. And so we're currently engaged in a larger study of a ketogenic diet in Alzheimer's disease, in which we're trying to do randomization. This is an NIH-funded study that will take us 5 years to do. It's called the Therapeutic Diets in Alzheimer's Disease, or TDAD study. And what we're doing is we're recruiting 80 individuals, and we're randomizing half to a ketogenic diet and half to a low-fat therapeutic lifestyle changes diet, which is really a heart-healthy diet. And they're on the diet for 12 weeks. And our endpoints are cognitive tests, so a clinical endpoint. And then we're including biomarker endpoints, which are designed to give us some mechanistic insight into what the diets do to our biology. So we're measuring depth of ketosis. We're measuring mitochondrial function, both directly in platelet mitochondria, and we're trying to indirectly get at mitochondrial function using magnetic resonance spectroscopy of the brain, in which we're measuring cerebral N-acetyl-aspartate levels. And we're looking at insulin signaling in the periphery. We're measuring lipid homeostasis, including ApoE levels. We're looking at inflammation markers systemically. And we're also performing a transcriptomic analysis in that we're performing RNA-seq on individuals on the different diets. So this is intended to be my summary slide here. And it kind of encapsulates a lot of what I've been talking about and tries to refine my thinking on the primary mitochondrial cascade hypothesis based on these recent data that I've been sharing with you. So the way I'm kind of looking at things now is that what happens is as we age within neurons, we see reductions in mitochondrial function. Perhaps these reductions in mitochondrial function are related to mitochondrial DNA, such as depletion of mitochondrial DNA, which eventually may reach a threshold that induces a retrograde signaling back to the nucleus and turns on ApoE expression. And when ApoE expression, which shouldn't be turned on in neurons, when ApoE starts to get expressed in neurons, either ApoE protein or the ApoE cleavage product then localizes back to mitochondria, where it may be a second hit on the mitochondrial function. So further worsening mitochondrial function. And if you are unfortunate to have an ApoE4, perhaps you're getting more of this second hit to the mitochondria, and that's what's increasing your Alzheimer's risk. So how would I treat Alzheimer's disease? Well, the one area we're targeting is here. We're developing drugs that will improve mitochondria and approaches that will improve mitochondrial flux to try and make carbon flow through old mitochondria in an old brain more like it used to flow when the brain was younger. So we're trying to address the carbon flux problem through mitochondria. And then we're also very actively engaged in trying to figure out what's mediating this retrograde signaling from the mitochondria to the nucleus in neurons, because I think that if we can figure out what's mediating this retrograde signaling, then we can figure out ways to inhibit it and then block this second hit on the mitochondria. So with that, I'd just like to acknowledge the many individuals that have contributed and do contribute to this research and to some of our funding sources. So with that, thank you very much for listening to me, and hopefully there's time for questions.
Thank you, Russ. So really nice talk. It's amazing from a fundamental all the way to clinical study. So they do have some questions here posted at the chat. The first from Elizabeth Park, Professor Park. Congratulations on this nice work regarding the keto diet. Do you think the effects are influenced by the diet directly effect on the brain, on the microbiome changes? Is the weight loss occurring?
Yeah, so so that's what we're trying to find out. Is it the if big question, if a ketogenic diet really does affect the brain in a beneficial way, is it because of the ketones in the brain? Is it because we're changing insulin signaling and insulin sensitivity in the brain? Or and yes, we're also interested in some kind of microbiome change. For example, short chain fatty acids are catabolized by certain gut flora into a ketone body. So one thing, and it's just amazing how this all becomes very relevant when you're planning these trials, because we were told by reviewers of our grant that we needed to give, if we're giving an MCT drink to people on a ketogenic diet, we had to give a drink to people on the heart healthy diet, and so we planned on giving a like a Metamucil thing, but we had to be careful not to give psyllium fiber, because psyllium fiber can be converted to ketones. So if there is an effect, I don't know what the effect's going to be. I mean, ketogenic diet has even been associated with reductions in inflammation. So, we've done a lot of studies now on how ketones affect carbon fluxes in neuronal cells, and they really do have impacts. Whether it's all due to carbon flux or whether it's affecting at a receptor, it's just hard to say. But some of these effects are truly protean. Yeah. So thanks for that question.
Thank you. The other question kind of on the same line from Eileen. So would the ApoE genotype be stratified in the TDAD study?
What's that
ApoE
again?
genotype would have stratified in the TDAD study.
Oh yeah. Well, we're ascertaining ApoE genotypes, but in the current study, and we should have enough power, or we should have the ability to kind of get a feel for, does the diet have more or less or impact in people depending on ApoE genotype. In the preliminary study, we couldn't really determine whether whether things were segregating along the line, whether responses were segregating along the lines of being ApoE4 carriers or not. But hopefully we'll be able to get some insight into that in the TDAD study.
Okay, thank you so much. I have a question regarding about the downstream of the mitochondria and the synaptic effect. You did mention about this. Can you speculate how that's work? So basically, so in your study, the clinical world did recognize they have a cognitive function improvement, and how that's work on the synaptic level.
Yeah, that's so we don't really, that I can't answer. We're getting the full MRS data now. I mean, you guys, I'm kind of jealous because you guys have 7T. So you guys can probably even measure beta hydroxybutyrate directly in the brain. And so, I kind of had to put together what neurochemical would I look for, to try and get some insight into that. But, NAA is a good one, I think, because it's really just looking at I think it's an indirect look at acetyl-CoA levels in your cell, in your tissue. So if you've got a lot of ketones, my guess is that you're going to have more acetylation going on. So hopefully you'll see more acetylated aspartate. But that's a good question about how to get a better view of synapses. I can see here that there's some other questions too. Let's see. This is one from Carmen. Amyloid beta induces AD mitochondrial function. Related to this, the MR spectroscopy studies demonstrated that changes in myoinositol, a putative glial marker, precede amyloid pathology, and is the most relevant MRS metabolite in humans, human tissue, animal models. Antemortem milliliter levels are
What's up, Jonathan?
myoinositol levels are associated with postmortem. Okay. So is it a mark?
It would be good because he did do a document.
I mean, N-acetyl aspartate, at one point it was hoped that might actually serve as a nice MRS biomarker that you can track on MRS, and it never, and people tried ratioing it, N-acetyl aspartate to myoinositol, and I guess it wasn't robust enough to really pan out. But I don't know, Carmen. I mean, who knows? I guess the question is what is amyloid pathology a marker of? So my best guess is that that the plaques are basically a marker of brain aging. So some people may deposit it and some people may not, but certainly that's certainly a big picture thing that's limiting the field, is that we're trying to base understanding a disease around the biology of a protein that we don't understand the biology.
The last question, Shankar asked about what are the roles of intermitochondrial lipid in stabilizing mitochondrial membrane?
Yeah, that's a great question. So we've sort of been barking up the tree of how mitochondrial does. Let me put it this way. Lipid biology is now recognized to be important in Alzheimer's disease. And to me, I don't see how you could divorce lipid biology from mitochondrial biology. So, so, the way I interpret that is, if lipid biology is perturbed, then mitochondrial biology may be behind that as well. We've looked at cardiolipin levels in some of our models of mitochondrial function. We've looked at lipid droplets in different models. I even have a grad student who's getting his PhD studying the mitochondrial fatty acid synthase apparatus, and it's just humbling. We're not finding differences in cardiolipin, although when you look at all those misshapen mitochondria in Alzheimer's patients, you might presume that there should be some problem there. But that one's a level above, grade above my pay grade.
Okay, sorry. The last question that's from Xinhua asked about the neuronal mitochondria, but the platelet and the neuron mitochondria, how that's compatible for the effect. So
this is really important for why I've drifted to this primary mitochondrial cascade hypothesis. So because the mitochondrial changes in Alzheimer's patients are systemic, either there has to be a systemic toxin that's causing the problem, or it has to be genetically mediated. And so that's why back in the 1990s, we postulated that the most of the three possibilities is it due to a systemic toxin? Is it due to systemic mitochondrial DNA, or is it due to a nuclear gene? we postulated it was going to be due to mitochondrial DNA because Alzheimer's disease mostly is a sporadic looking or pseudosporadic disease. So in mitochondrial DNA genetics are perfectly situated to cause sporadic or pseudosporadic disease epidemiology. So basically in 2021, what I think I'll just share with you basically what I think is happening. I think in certain mitochondrial DNA haplogroups, like haplogroup J, there are variants in the mitochondrial DNA control region that control mitochondrial DNA copy number. I think that's what we're picking up on in the cybrids. We're picking up on variants in the control region that determine mitochondrial DNA copy number. And when you have lower mitochondrial DNA copy number, you see lower cytochrome oxidase activity and a lot of these other changes that we're detecting. And it puts you on in terms of how close are you on the proverbial cliff to getting Alzheimer's disease? If you're starting with a lower mitochondrial DNA copy number, presumably you're going to be closer to that proverbial Alzheimer's cliff than if you're starting out with a high mitochondrial DNA copy number.
I really appreciate it. I guess we're already passing 5 minutes. I guess in the future, it's worth it for you to visit Mizzou for the continued discussion. Really appreciate it, Russell.
I can guarantee you we will have Dr. Swerdlow back to visit Mizzou some way, somehow.
Okay, really appreciate it. Thank you.
Hey, Russ.
Guys.
I'm sorry.
Good to see you, Russ.
Good to see you all.
some
Thanks,
questions
everybody.
cannot be answered.
Bye-bye.
Thank you, Russ.